So, good afternoon, everyone. Thank you, sir, for accepting our invitation. So, actually, our head of the department planned this general club because students are more interested towards such activities. So, they actually initiated this and they requested head of the department. So, we wanted to inaugurate with you and with your lecture because we know you motivate students very well. So, we wanted you to inaugurate this general club. And sir, before going to that, I wanted to just briefly tell you know very well about this, but few students who joined recently, who doesn't know what is our department, or what is the strength, and what activities we have been doing. So you can see here areas of research we are working on, cancer biology and neurology, neurobiology, as well as biocatalysis, protein engineering, cardiovascular biology, and computational, as well as structural biology, and pharmacology and toxicology, as well as stem cells and regenerative medicine, and immunology and infectious diseases. And workforce and projects. Sir, this is a total workforce we have. Our scientific staff is, so our scientific staff is total 21, and temporary scientific staff 4, as well as technical staff group 3 6 members, and group 2 3 members. PhD students we have 68 total. So that's why we wanted to initiate this general club. So they are more benefited because of this. And project assistants 29, and dissertation students 48. Contract staff 12. It's almost 190. Total staff is 190, and ongoing projects we have like total 29 as PIs, and including CSIR and GAP projects. And as a co-PS, our department has total 15 projects. Yeah, again, the man who needs no introduction, but still I wanted to give brief introduction to the new joiners who joined PhD recently. So this is a brief background about Srinivasa Reddy sir. And he did his PhD from University of Hyderabad under Professor Govardhan Mehta. And then he attended a postdoc in 2 universities like University of Chicago, followed by University of Kansas, with professors like Sergey Kojmin as well as Jeffrey. And total he has 25 years of research experience in natural products as well as medicinal chemistry. And he has been serving as a director for IICT, we know this. But addition to this, recently he also worked as a director for IIM Jammu and acting director for CDRA. But addition to IICT, he is now also acting as an acting director for AMPRI, Bhopal. And still with that busy schedule, he allotted some time for us. So we are lucky. And one of his inventions, so went to phase 2 clinical trials in collaboration with Novartis. And this is again brief recognitions and awards. If you see, he has many awards, but among the best ones are like J.C. Bose National Fellowship as well as Sanswarup Bhatnagar Prize. And he has many fellowships, and we cannot list here. So the 2 important things are like Fellow of Indian National Science Academy and Indian Academy of Sciences. And he has total 145 publications with and 35 patents. Among 35 patents, 2 out-licensed. And he has most of his publications in American Chemical Society journals, like all prestigious journals, JMC and OL, and many other decent journals, decent publications. And yeah. Yes, sir. With this, thank you, sir. Sir, please. Yeah, sir, we want you to inaugurate this.
I don't think required, mic required. Let's just stop it. Yes.
Yes.
So I'm inaugurating now.
Yes.
Yeah. Good. Congratulations to all of you. It's a new initiative, good initiative. Let me see where it is. Yeah. Okay. First of all, thank you, your organizing team, both Srinivasa Reddy and our leaders. Thank you for making me part of this inauguration or part of this journal club. I think it's a good idea. I think I appreciate. And also I am a chemist by training, and chemist coming and lecture in biology. Actually, in fact, that is good. I'll tell you, this is one of the best place, IICT, where we have chemists and biologists together. But I did not see that much of collaboration what we expected. So hopefully, maybe some individual level we are doing some places here and there, but I think I expect more collaborations between chemistry and biologists. So that's, I think we need to do this ideal place, basically make compound in one lab and bring it and give it. This is like industry. I can tell you most of the industries, I worked in drug discovery industry, this is ideal place. I think place like this is, you are fortunate to be here. I mean, like you mean like our all scientists to be fortunate to be in this kind of environment. We have one of the best facilities in terms of chemistry or biology. So if we cannot do together, I don't think any other place in our country can do it. I think this is I can tell you that this is one of the best places. Since I've roamed around the country, I know many places, many labs. So our IICT is ideally suited for collaborations, particularly chemistry and biology. So hope we'll continue to do further better and better in the future. Okay, today I'm not talking as a director, I'm only talking as a scientist. Okay, so in between I need about 5 minutes time. I already indicated to Dr. Sisla, I need to make one call around 4:45, okay, 4:40. Okay, so, okay. So since students are there here, I put couple of slides on how this drug discovery works. Since I have little experience in drug discovery process, so I thought I'll put you, like basically, I'm sure many of you are aware of this one. I'll kind of refresh you how this drug discovery process works. So for any professionally driven drug discovery companies, what happens is first you identify disease or target, some of those things. In the beginning, you'll have some idea. So that we have to do kind of through evaluation of our inputs from the literature, from the committees, from the experts. You get some information. Based on that one, you find a target or indication what you want to do depending on the demand. Again, business point of view also you have to see. So once you identify target, then you form a team. Again, team comprises of chemists, biologists, regulators, and other pharmacokinetics and different departments. They put together, they make a team. They form along with, there's a project manager also will be there. They will form kind of a flow chart, how to move forward. So that's, they put some kind of target product profile, we call it TPP, we call. If somebody who worked in industry, they'll understand better. TPP, target product profile. What do you want to achieve at the end of your project? That's what they set the goals, then they put filters. And once you are ready with the plan and everything, then chemists go to bench and they make standard compounds, and similarly biologists go to bench and they set up assays using known assays or known ligands or whatever available information. You put that one using the standard chemicals. So once you are through that one, then you chemists go to bench and make molecules. Make molecules. What is the difference between earlier molecules and present molecules is, earlier molecule they make standard molecules which are basically which can be used as a standards, and actual molecules, what that means is patentable molecule we call, which are proprietary molecules. Like any company want to put money into this, and because it's a big long process, lot of investment goes into this one. If you don't have a proprietary, if you don't have a patents, there is no use of putting effort in this direction. That's where you need lot of like a clarity on this patentability of molecules, particularly if you are working in industry. If you are working in academic institutions, still for publication it's okay, but for industry, you cannot afford to lose that one. So that's why they are very, very careful in making molecules which are patentable. So once you are through this one, then again, chemists, biologists work together. You go through all this in vitro screening. There's a lot of medium throughput, high throughput screening. All the screens are available, robot screening, depending on the company size, you get there. Once you are there, then you go to in vivo models. Again, I don't have to tell you people what kind of models, and depending on the indication, you go through that models. Once you are through this one, then you go to safety studies, including initially you do this acute studies, and then you do long-term studies. We have to do different species. You need to do 2-3 species we need to do. Once you are through this one, once you are clear all these hurdles, then you go to regulators. That is the IND application we call it. So we go to regulator. Regulators means like a US FDA, like Indian equivalent. What is Indian equivalent? DCGI, Drug Control General of India. Like Europe also something else like that we have. So we submit the document to them. So once then they have a committees, the sectional committees will be there. They'll go through the document in 30 days, generally 30 days actually. 30 days, if you don't have any problem, then you can go for dosing in humans. So that's where you get clarity in a month time. So then you go to phase 1 clinical trials. Phase 1 clinical trials is only they get information whether compound is safe or not. Although we know in animals the compound is safe, otherwise you cannot go forward. So in phase 1 you take healthy volunteers. Why do healthy volunteers? In particular male people they take. So that's kind of generally that is preferred one. I think that's what they do is most of the cases because of hormonal changes and other problems. So there you do single ascending dose, then multiple ascending dose. That's basically easy to remember, SAD and MAD, SAD and MAD. So you do all that and finally you get the dose which is safe dose. So once you are through this one, then you can basically from that information pharmacokinetics you get, what is the safest dose you get from the phase 1 clinical trials. So once you are through this one, then you go to actual proof of concept you get in phase 2 clinical trials. Phase 2, there you recruit patients, then they do limited trials we do. Particularly if you are going for diabetes, means you recruit diabetes, you said that what HbCN level, all these parameters you set up, and also you set up that markers, what you are going to measure that also. All those parameters you set and you go to that phase 2 clinical trials, you do that one. There, first time you are going to get whether drug is working in humans or not, you will tell. Before that, we know some information from animal models, but the first time you get the information in humans in phase 2 clinical trials. Phase 2 clinical trials, once you are through this one, you are satisfied, then company or organization goes to phase 3 clinical trials. Simple terms, phase 3 is nothing but scale up of phase 2. Like multiple locations you do this one, and phase 3 and different populations, different ethnics also they do that one. Then you finally phase 2, phase 3, you get the information how efficacy your compound, then all these results. Then again you submit to regulators. So there you submit to regulators, again same regulators. There also you have all the committees, expert committees will be there. They evaluate all the data and then they give the approval. So once you give the approval, then you go to clinics. So this is what the general process, what we do in general drug discovery process. If you want to discover a drug very systematically, right? But like out of all, drug discovery is the most important thing, because if you can find a best molecule, or if you can select the best molecule, you have a high chance of success. Otherwise, you see how much money into go, like lot of years of time and also a lot of money goes into this one. That's going to be waste of money, right? That's why you have to be very, very careful in choosing molecule in the beginning. So we generally use lead character. What is lead? Somebody will ask you, because we all the time we use hit, lead and all that. Sometimes we get confused, we mix up. And as such, there is no definition, but in generally these are all some of the numbers followed. Again, depending on your target and other things, it matters. Also, these numbers also vary little bit. Look at this potency around 100 nanomolar. Again, liver microsome stability is about less than 1. and similarly ppb, solubility, c-log-p, molecular weight. These are some of the parameters. Hurg liabilities also, anything is there, if it has to be more than 10 micromolar. These are the parameters you do. In addition to that one, again as I said in the beginning, there should be clear scope for the freedom to operate as for patentability wise. So then only you can call that particular compound as a lead. So many times people use lead molecule, but unless you have some of these characters, you can't call legally lead molecule, right? So then the other, before candidate selection, there are other parameters you can get into. Again, I'm sure you all know about it, just like metabolic rates, metabolite profile, in vitro glutathione conjugation, and PK, bioavailability, formulation, dose response, and all those things we need to, again, PK, PD, pharmacokinetics and pharmacodynamics correlations we need to see. We need to go through all this. If it is a brain molecule, if it is a CNS molecule, then you need to worry about your brain exposures, all that we need to do, right? After all these things, you find a candidate. Again, some of the other parameters also you need to go through this one. Again, I will not get into this, but so how do we start? So these are all we know. Now drug discovery process. How do we start? We need to have some starting point, right? So one of the starting point one you can think of is library screening. You take large libraries commercially available. You start from that. You can first to get some kind of hit in the beginning. So you go through this one, and many commercial libraries are available. Even we did purchase in our mole bank. Some of them we have, and another one is analoging literature leads. You can also take something already known in the literature. Do kind of if it is a patented molecule, try to come out of that patent by making some kind of derivatives. Those things we can do. So that is another one. Of course, that is going to be little challenging because of the patent space. And again, analoging ligands and substrates. In our body also we have some of the substrates. You can also analog those things. And another one is one of the best approaches, structure based drug discovery. Particularly this is very useful if you have a crystal structure available. So that's kind of best practice, where really you can visualize and you can see how this your compound is interacting with the different like how your pharmacophore interacting with your all the different amino acids, right? So that's one of the best method if you have crystal structure. Even then there are other way to do it is you can also develop some kind of pharmacophore model or homology model you call it. So based on the information also you can create that models. So that is less reliable, but according to me something if you have crystal structure, there is ideal way to get started from there. And another thing is there is also another way to do is so medium throughput screening, what we call it as a small like a target based libraries, natural products, and screening of fragments also we can do. These are all very successful in this way, where you already have seen some kind of like at least based on the literature information, more success comes from these kind of areas. So that's where I'll spend some time on this. Hey, where is slide here? Let me put my
yeah, laptop separate.
Compatibility. You have this. You have this converter.
No, sir, we don't have this.
I may.
We have both connections, HDMI and
No.
This you can use a converter.
We don't have, sir.
Aye. Yes. Oh, that's okay. Give me some. Okay, this was the slide, right? Okay, in my research group, of course, I started my career at NCL Pune. So since I have little experience in both medicinal chemistry, and during my PhD, I worked on total synthesis of natural products. I did. So based on that experience, we started this working on total synthesis and also applications. So what we do is in our group, drug discovery programs, of course as a medicinal chemist, and also we design synthesis molecules and lead optimization we do, and finally we'll get into as application oriented, different things we do it, but majority work is towards drug development we work. So that's what we do in our lab. So why again natural products, why it is so important? Because if you look at our medicines today, what available in our medical stores, more than 40% of them are related to natural products. And either directly or indirectly, some of them became as such drugs, and some of them derivatives, some of them are basically they converted into different derivatives, analogs they made from that, or from that inspiration also they took from scaffolds. So these are the different ways they used the natural products. So why again, particularly just to give some justification, excuse me. So scaffold diversity, structural complexity, and also they typically have higher molecular weights, mass, and larger number of sp3 carbons. That also tells us that basically that particular shape, like if synthetic compounds mostly flat, whereas natural products they have this more of three-dimensional structures. Specific centers are there, chiral centers you call it as mostly. So that is the advantage, and larger number of oxygen atoms and fewer like also nitrogen and halogens also there, and higher number of hydrogen bond acceptors and donors, and lower C-log-Ps, higher hydrophilicity, some of the parameters based on the information we gathered here. So right, just to give you a little like example, one of the examples which why I worked in industry when I was in industry in Novartis along with Advinas Pune, we worked together. So one of the inspiration came from this compound called fluorescein. This was originally isolated from this bark of pear and apple, and this is original compound, and there are also some problems with the hydrolysis and other things. Then finally through modifications they came up with, again it's not Advinas or Novartis work, this is literature, but I will tell you where we contributed. So this is T1095, one of the lead molecule at that time, and again issues with this selectivity, and also this is called O-glycoside we call it. This is a sugar, right? The sugar linked with aromatic compound with O, that is called O-glycosides. In our body we have glycosidase enzymes that can cleave that one. So to address that issue they came up with the C-glycoside. Basically they removed this oxygen, they came up with this particular compound, they call C-glycosides. So these originally they showed anti-diabetic effect, but later they found that this was going through SGLT inhibitions, that is a sodium glucose co-transporter inhibition. This is one of the beautiful mechanism right now available. So made this compound C-glycoside, that is called dapagliflozin. Similarly canagliflozin. So these are some of the molecules came up during when I was working in Advinas Pune. During that time there are a couple of other molecules also going. Then we discovered a molecule that time we used to call this as a LIK066. This again very C-glycoside related compound and patentable compound, and again later this named as a licogliflozin molecules. So right now it is phase 2 clinical trials just completed, and also this is also being tried for NASH and PCO patients. So this is a molecule what we discovered in my research group where I was leading that project as from chemistry side. Of course biology side another project leader will be there. With the help of chemists and biologists we could identify and took it to clinical trials. This is a kind of satisfaction where we design molecules. It's a beautiful even PKPD we used to see this molecule. Basically you measure that, you take the urine and measure the glucose level. The kind of SIR we did, that was fantastic. Hopefully someday we will write that paper. Still is active trials, that's why we are not writing that paper. So that's one of the good contribution from our group that time. Right? Yeah, just a few minutes. So here is a snapshot of molecules what we synthesized in our research group over the years. It's most of them are for the first time we synthesized in our lab. Then we collaborate with the biologists like you, and we create value for those molecules. Look at this one, the structures are very, very different, and also this activity is also different. Actually, as a chemist, I don't really worry about which activity and other things, but as long as it is active, and if I can find a suitable collaborator, we make those molecules, we collaborate with them, and we take it forward. We again, depending on the interest, we'll further optimize. So look at this, some of these molecules, this anti-proliferative activity, anti-malarial, like this TRP modulation, and some of these glycosidase inhibitor. Look at this one, some of these molecules, anti-inflammatory. This is one of the major project in my research group. It's almost going on for more than 10 years. I'll tell you that story now. And like this, these are the molecules we have in our hand right now. So give me just 3 minutes. So there is a parliament session is going on. We get some questions. We're supposed to respond to a few things. Okay. Okay, yeah. What I was telling you, these are the molecules we synthesized in our lab. So why do you want to make these molecules? Because most of the time nature gives isolation, we get only small amounts. But if you make these compounds in our lab, we can use it for biological assays, and also we can also derivatize, we can make analogs, and we can come up with a new scaffold. That is the advantage with this this synthesis of these molecules, right? So I'll tell you a couple of stories what we have. So I'll be crossing this time, is okay, right? Okay. So This is one molecule. This is cladosporin. This was originally isolated from fungal source in 1971. That was my year of birth. Okay. Then you may ask, why are you interested in that old molecules? Because recently they found a target for this molecule, this very potent anti-malarial compound. It works through lysyl tRNA synthetase. So that's one of the reason we became interested. Although synthesis is known, actually, in fact, Dr. Mahapatra also made some time ago, 2013, this compound. It's a synthesis. So most of the people, they synthesis and they leave it, that molecule. But my interest is slightly different. I make molecules and do more derivatives, more analogs. I collaborate with biologists. That's what I do, slightly different from some of our other principles. There is a 2 to the power of 3. So that means 8 compounds. So 8 possible isomers are there, right? Then we thought that why don't we make 8 compounds and come up with the best one? So that's the idea we started. So we thought that we'll make all possible 8 compounds instead of making 1 compound. So we thought by changing, look at this, all look same, but actually they're all different. All are different compound, different isomers. You look at, both are up, both here, these both are down. So all isomers, all possible 8 isomers we wanted to make and put them into biological assays, like then come up with the best compound from this active stereo isomer. So what we also coined here is SAR. You all know about it, right? You heard about it at least. Structure activity relationship. So what we did in our group is, there is something called, we added SSAR. Stereo structure activity relationship. That's what we are trying to coin that word. So stereo structure activity. How the stereochemistry also impacting your like binding and other parameters. So that's where what we did is, again, I'm not going to go through the synthesis. We took these fragments, we went through all this known knowledge in the literature, using our knowledge and other things, we made these compounds, right? After making these 4 compounds, then we also made, if you change the stereochemistry of one of the starting material, you also get other isomers. And prove the structures without any ambiguity, because this is proving structures also a very tough challenge, because all look same, NMR very similar, so very challenging. That's a big exercise for students, it's a very tough job. So one student joined with me, Pranay, he from IIT Madras. So I gave him this task. Initially he was reluctant, but later he really enjoyed this project and made all the compound, how the difference and everything he analyzed. So crystal structure also available. Basically we crystallized, we made sure that whatever we made is right. We compared with the opposite isomer instead of our activities because of the Professor Padmanabhan. So we collaborated, then we took entire library of that 8 compounds, and using this thermal shift assay in the beginning, then we did assays, then for further parasite growth inhibition assay and enzyme inhibition assay, and finally come up with the best isomers. So what we have is, these are the results. So what we did is, we took all the isomers, all the compounds, put it to these assays, whatever I discussed. In addition to that, we took all the compounds separately, each compound, we basically incubated, we co-crystallized with the Plasmodium enzyme, KRS enzyme. So what we found is 2 compounds got co-crystallized out of 8 compounds. In fact, those 2 compounds only found to be the most active compounds. We basically segregated out of 8 compounds, 2 compounds as a class A and another 3 compounds as a class B, depending on their activity. So what we found is co-crystal structure, whatever co-crystallized, those 2 only the best compounds. So from this, what we found is stereochemistry in drug discovery is important. Not that we are doing first time this one, but using this particular scaffold, this particular enzyme, we proved once again that the stereochemistry is very, very important, and we are trying to show also this SSAR, stereo structure activation, which is important. So what we found is this particular methyl group here, this particular methyl group, whether it is up or down doesn't make difference. Otherwise, these other 2 centers have to be essentially that particular stereochemistry. Basically, this 3R, 10R has to be, that particular stereochemistry has to be fixed, but whereas that methyl group can be either up or down, either is fine. So that's what we try to give that information to the literature, right? So after that, what next? Then after doing a lot of other lead optimization, we found the molecule called CL2. So this particular one, when this is a optimized lead so far now. So the difference between the starting point and this is addition of this particular group. Although we made about 30-40 compounds, but finally we came up with this conclusion that this is a compound, best compound in our hands right now. So all other parameters also we measured, and we found that basically from when moving from original compound to this lead compound, we increase the drug likeness component. Some of these parameters, what you can see here, some are up, some are down. So basically increase the druggability of this particular compound. So we need to go forward. That's where we are at this stage. Right? Then I'll tell you another important project from my research group, that is solomonamide. It's a potent anti-inflammatory compound. Again, this is structure looks complex. That is also challenging for chemist students to make this compound. Again, why it is important? Because we looked at the potency of this molecule. It showed about 60% Pa-edema reduction in mice, originally isolated compound. That's 100 microgram per kg IP dose. So that's what is reported in the literature when they isolated this compound from marine sponge. So we became interested, because this kind of activity you see, you need to use dexamethasone, maybe 10 milligrams you have to use to see that kind of effect. So again, I don't have to tell this audience what is important. Inflammation. Inflammation, if that is a protective, we need, it serves as a protective function, but if it is uncontrolled, you get into all kinds of diseases, right? You can think of all kinds of diseases. Almost every organ can get affected, lead to different diseases. That's why if you can control, if you can have good anti-inflammatory compounds, then it will be a good way to move forward. That's where we became interested. Again, this is some of the literature we published. We have been continuously working on this group, but I just tell you what exactly we did. So some of you who understand little chemistry here, see solomonamide B and A, the difference between these two compounds is the hydroxy is not there, hydroxy is here. So that's why we designed a compound which can give both. There are some of the reactions which selectively we can do oxidation here. You can get this B selectively. If you can do dihydroxylation, and if you do selective benzylic oxidation, we call next to benzene group oxidation takes place. That is the benzylic selective oxidation. Then you get to solomonamide A. That's what we designed, and some of the strategies we followed, what we wanted to make. Finally, what we did is, we synthesized all the compounds, not only that single compound, we made library of compounds, around 40 analogs we made. All these macrocycles. After making all the macrocycles, then that time I was working in NCL Pune, then I collaborated with Dr. Manoj Batwal, CDRI. We tested all these compounds using this Pa-edema model, and we found that a couple of molecules are looking better. So then we wanted to do, so basically what we did is, we started from here, and after making analogs, then we come up with that particular compound, which is simplified compound. Look at this on the structure, there is too many stereocenters. We came up with a simplified compound, and which showed as good as original compound. That's where we came up with this particular compound. And we wrote some beautiful story in this recently, one of the paper, this in biology, JBC we have, right? It's like JBC, it is a JOC, Journal of Organic Chemistry we call. All efforts we put it, it is like a 45-page printed document. Like very rarely you see that kind of big papers. It's a huge story, because last 10 years story we put there. So again, as part of our group's interest of stereo structure active relationship, what we wanted to do is, see, look at this one, there are two stereo centers. Again, 2 to the power of 2, you get 4 compounds. You can make 4 compounds. Right? What you want to do is 4 compounds will make. In addition to that one, there is a double bond also there. You can also reduce double bond, you can also keep the double bond. 4 plus 4, 8 compounds. Let's make 8 compounds, we'll get into the same similar assays, biological assays we do, and come up with the best molecule. That's what we wanted to do. Again, we started from here, and we made this, all the 8 possible compounds, and some of them are crystallized, and we made sure that structures, whatever we synthesize is right. Once we are there, then we screen these molecules. Again, with Manoj Batwal, we did these 8 compounds. We found that out of that, there's a 2127, 2129 came out to be the better ones when compared to other stereoisomers. So we took those 2 compounds, then we put it, we also checked pharmacokinetics of these molecules. So generally peptides are not that good, like they don't show good pharmacokinetics, whereas we are lucky to get that both compounds oral bioavailability, one is the 70%, one is the 90% oral bioavailability, right? That's where recently, of course these are the things recently happened, and once we, total synthesis, we made this one, SSIR, we got to this particular compound. Now we are collaborating with Dr. Sai Balaji with this particular compound, one particular compound. Then we are trying to do some of these models. So I'll show you one particular interesting is, where is your name? Your name is somewhere here. Okay. It's visible, right? Yes. Yeah, okay. So what we wanted to do is some of this DSS-induced ulcerative colitis induce, and you can measure some of these parameters. Again, you all know what are these parameters to measure, and what we want to do is two compounds we take, two different doses. One is 25 mg and, sorry, three doses, 25, 50 and 100. These three doses we wanted to do. So we did some of these experiments. We have some interesting results. I'm not going to show all the graphs, but something visible I can show is something like this is something interesting, which hopefully will be able to convince that we have something interesting. So this particular one of the compound, we look at this one. This is a normal one. This is colon length. That's what I was told, that this measurement is one of the indication. And if you look at this one, this particular compound 2129 with 100 mg, almost close to your normal one. So that's something or Dr. Sai Balaji and Dr. Sisla are very excited. Like at least they think that is a potential to move forward, and that's where we are there in this one. It's a beautiful story. We started from, of course, 10 years back, we started this work, making macrocycles, natural product, and a lot of chemistry, interesting chemistry we developed and simplified structure. Then we did this SSIR, stereo structure, active relationship we did. Then I moved here. Then I got connected with our group. Then we came to this stage, and we are excited to move forward in that direction. Then I'll tell you another story. So I'm telling some of the stories so that some of you can also see whether not only with my research group, other research groups also, there are also interesting compounds are there. Best way is to talk to them, discuss with them, and get collaboration. That's the ideal way to move forward in the future. Right? This another interesting molecule we picked up is hunanamycin A. This showed 4.8 microgram per ml, the Salmonella enterica. And if you look at the structures, they look very similar to riboflavin. And so why is interesting? Salmonella interesting is which causes food poisoning, and some of the, at least numbers shows that foodborne disease caused 600 million foodborne illness, and about 4.2 lakh deaths annually occurs. That's available data, but I'm sure underdeveloped or developing countries like us, these numbers will be much more. We don't have the data, but there must be numbers are more, right? That's where we became interested in this molecule. We also look at, when we pick up the molecules, natural products, we also look at their, how druggable that molecule. In medicinal chemistry, there is something called principle, Lipinski rule of 5. If your compound follows that particular Lipinski rule of 5, these are the parameters. Molecular weight has to be less than 500, C-log-P less than 5, hydrogen bond donors less than 5, hydrogen bond acceptor less than 10. If you follow that one, there is a good success of making oral drug. That's where, wherever follows, we pick up those molecules. Not that we essentially follow, but if the molecule follows in that line, we pick up those molecules, we focus on those molecules. So that's where we calculated, we found that this is one of the good starting point. These are some of the parameters. So what we wanted to do is we wanted to synthesize first, because this compound originally isolated from marine source, like marine bacteria source, they isolated 0.8 milligram, less than 1 milligram they isolated. I don't think we can do much with that compound. So that's where we wanted to synthesize analogs and lead optimization. That's what we wanted to do as part of our research group interest. What we did is, we did couple of synthesis, but I'll show you one scalable synthesis. What we did is, we took this riboflavin and we did some of the transformations. We got this particular intermediate. Then we did with the phenyl bromide, we get to this particular compound. There is something called Friedel-Crafts alkylation. If you do, then you get to this compound. So original isolation 0.8 milligrams. So now we can make more than 10 grams within a week time. Within a week or 2 weeks time, we can make multiple grams of this compound. So that's the beauty of synthesis. Actually, during like some of the trainees comes, dissertation students. So I give it to them some of time this project to get training. They make this compound. Basically, you don't need really expertise in synthesis. So that's where we optimize these conditions. Anybody can make this compound right now. So that's where we have like a 3-step sequence, no column purification. Overall it is 48%, 96% purity. Basically while washing only we can purify this compound. That's what we synthesized. We can make multiple grams of this compound. Right? What next we did? We also wanted to understand mechanism of action, how it is acting. So since it is related to riboflavin, so initially we thought that this is probably going through the mechanism called riboflavin synthase inhibition. Riboflavin synthase we don't have in animals or like humans, we don't have, we get from food. Whereas some of the bacteria, they have to synthesize, they have this riboflavin synthase enzyme. If you can inhibit riboflavin synthase you can selectively kill the bacteria, or at least you can harass the growth of that bacteria. That was the advantage with this particular target. That's where we became interested. We wanted to see whether it is really going through riboflavin synthase or not. Again, I collaborated with one particular group. So again, I don't have any boundaries. I go to any place, but I connect with the right people and get them tested. So then I connected with this particular group, and what we found is we incubated with this in E. coli, riboflavin synthase. What we found is this is a decreasing concentration, increase the production. This is a penultimate step of the riboflavin synthesis. So you can see the beautiful dose response you can see. So you decrease the concentration of hunanimycin, that increases the production of riboflavin. That basically we try to show that it is going through riboflavin synthase inhibition. That's a beautiful dose response we have, right? So once we have that target information, some other information, then we synthesized more than 70 compounds around this compound. Right now we have a compound which is 8-fold more potent with the original compound. So this is 548 we call it, and these are the right now what we optimized compound. This is also we checked PK and also no toxicity issues with this particular compound. More than 2 grams, it's no liabilities. And in addition to that one, since it is interesting compound for salmonella, we are also looking as basically exploring as a food preservative. Can we think of food preservative? It is a safe molecule, and again, it is like a riboflavin, and how safe we know that one. So that's what we are thinking in that direction as well, right? So a couple of other things, just I'll show you a few things where some of you can also utilize or use it, or you can think of using this compound, right? We all know cannabis, right? Everybody knows, right? Yeah, ganja, right? So there are molecules like a CBD, THC. There are some of the two compounds are very, very important compound. They became drugs also. Medical use also we can use. So this is CBD. This is CBD. Of course, this is dimer. That's what isolated from this group. Very few milligrams they isolated, and I think they used about 500 kg of this cannabis material. They isolated 2 milligrams of this compound. 500 kg of cannabis, right? Where do you get that one? So that kind of, again, one way is you can also synthesize starting from this CBD if you have, that you can isolate from this cannabis plant, and you can dimerize and you can get this compound. But access to this material also challenging, and very few places we can do it. Of course, we have, when I was working in IIIM Jammu, I got interested in this cannabis research, because they have a permission to do that one. They grow. So like all plants, like they grow that one. In fact, there is everywhere you see outside the roads, and the roads also, side by side, roadside also you see that one. In fact, in IIIM also, there's a lot of weed, this particular one. So people use that one for isolating molecule, not for other purpose. So again, challenge is this was isolated a couple of years back. So then we became interested in this molecule. Since it is difficult to access this kind of material, and also CBD also problem, at the same time getting access to that cannabis also problem, because 500 kg can give only 2 milligrams of this particular compound. And also it has some interesting biological activity. I don't know whether I put it something like a TRP modulator. So that's some interest on this one. So we thought we can make this compound. It's chemistry wise interesting, and also biology also something interesting. Right now what we have is, very recently what we did is we took this starting material, we went through this again. I'm not teaching chemistry here, but we made this compound cannabidinol. So I already got a couple of We just published this paper, couple of requests that they want some material, this one. But our biologists are, nobody is interested now. So we made this compound, and right, in addition to that, as part of SSIR, we are also making some of the analogs of this compound. So right now, we have few molecules. Again, this is all not explored. No biology explored with this molecule because nowhere available these compounds. Right now, only we have these two compounds, except that who isolated, they have 2 milligrams. Now we have this compound. So some of these compounds are available here with me. If you are interested or something interesting in your domain, you can also look into that. Okay, that's one thing. There's another interesting compound is this nitropuricin. So this compound, again, this a few years ago it happened. Again, these compounds are sitting with me. You can also see, just I'm trying to advertise here, okay? If you are interested, you can see. So this is something interesting compounds. They showed a lot of activities with this compound. Of course, one particular one, I put it here, antiviral activity, but I'll show you the next slide. So they filed almost 15, 16 patents, Chinese patents. Look at this one, this rhinitis. They used all these models. These are animal model data I'm showing you. These are for rhinitis and also oral ulcer and CHF. They're all animal data. If I have to believe that the patents are right. So they also compared with all this captopril and other things. Then also you can see acute renal failure, renal fibrosis. They made this, basically they tested in animal models and they all filed patents. Nobody published. But our group, because after seeing all this activity, my interest is to make this compound analogs and connect with biologists. So right now, what we have is we started with this one. We synthesized. We have now this many compounds, about 36 compounds with me right now. So you can also think of this connecting, whether somebody can connect with this one and make use of these molecules. So I'll be happy to connect with them. So this is what right now we have, these compounds. Right? Again, last few minutes I'll take. I also put in my title that silicon, right? I want to tell you some story about silicon. That's another interest in my research group. So you all know this is a drug discovery process. In the first slide, we started with this biological target. We go through all this process, lead identification, lead optimization, preclinical and all those things. But most of the time we spend time in lead optimization. Why I'm telling is, so if you want to see, if you analyze some of the major discoveries or major drugs, they synthesize almost more than 5,000 to 10,000 compounds you need to make. And they go through these approaches like analog synthesis, primary, secondary assays, all these counter target assays, in vitro, in vivo, in vivo pharmacology, in vivo admit, all these things we need to make. This is a conventional approach, right? This is what we do. And we require about 10 to 12 years and a lot of money, more than 2-3 billions you need that money if you want to get into the market drug. So that's kind of traditional approach. But recently, I'm sure some of you must have noticed, there is something called a deuterium switch. So what they did is they replaced hydrogen with a deuterium. Like hydrogen has either isotope, right? Deuterium. When you put deuterium, you can also modulate pharmacokinetic properties. So that's where in this approach, what we get is when you replace deuterium with, if you replace with hydrogen with the deuterium, so you can modulate, depending on the SIR, you need to put that group. What our group is interested is, can we replace carbon or sulfur with silicon? So again, we are not the first time doing, but in India at least we are trying to do that in my research group. What we do is, deuterium is known in the literature, or already marketed drug. What we are trying to do is Silicon-Incorporation. Can we incorporate silicon? Of course, silicon is, our body doesn't make silicon. So nowhere, no amino acid, no compound in our body has silicon. But can we use that silicon here? Because as such silicon, there's no serious problem, at least what we know from the literature. So we thought, can we use that one? Again, some of these parameters when we picked up these molecules, they are the parameters, basically basic logic behind what we use, because when we talk about if you want to convince others, largest covalent radius, no intrinsic toxicity, increase in lipophilicity, access to compound that do not have stable analogs, and more electropositive. In addition to all these things, it has IP. You can get patent protection. So I'll tell you one or two examples how exactly you can use. So this is haloperidol. Instead of silicon, if you have, that is called haloperidol drug. So by putting, what happens is in case of carbon, that get dehydrated, you get olefin. Olefin means double bond you get. That double bond is a mutagenic. So by putting silicon there, that you will not be able to make the double bond. So that's basically you are arresting that mutagenic compound formation. So that's something you can address this by metabolic pathway. And similarly, compound, look at this compound, this carnitine. This particular compound has more penetration towards cancer tissues by putting silicon. That's what is proved in the literature. Similarly, if you look at this particular compound, there is something called transition state mimic. So generally we believe that in our body, if you have a carbon molecules that can go through this transition state mimic, we call it as two hydroxys on the same carbon. That is difficult to make in real time. Although we propose intermediate, but that is difficult to make, difficult to handle that compound. But if you put silicon, you can make real compound. So can we use that kind of concepts? That's already known in the literature. But what we did in our group, this is one of the contribution we did is linezolid. What we incorporated is silicon. I'll just tell you what exactly. Of course, we have been doing like last more than 10 years, we have been working on this area, and we filed several patents and published papers and filed patents for different things, mainly of course CNS related diseases. Again, I'll tell you what exactly we want. I'll give you one example, you understand better. This is a linezolid molecule, right? This is already known for various treatments, various infections, available as a tonic, ointment, everything is available. Syrup, tablets, everything is available. Okay, similarly, look at this other molecule called sutezolid, that is for treatment for TB. The difference between these two compounds is only difference. Oxygen and sulfur. We looked at all the SIR public information from patents and publications. We found that if you put some other groups, that is okay. I mean, like that will not give you any problem. And also sulfur size also we looked at that one. But then we came up with this crazy idea. That time we thought is that a crazy idea. Can we put silicon there instead of sulfur? Can we put silicon and remaining all the groups keep it as such? So we call it as the original molecule is linezolid, then we called it as a silinezolid. So can we synthesize? What is our expectations? Because we are not changing too much into the structure, only sulfur is replaced with the silicon. It is expected to show similar activity, if not super active. So that's what we wanted to do. In addition to that one, it is also expected to have better PK parameters. Why I'm telling better PK parameters? When you look at this molecule, this morpholin, in cytochrome per 50 oxidizes this next to oxygen that forms lactone and cleaves off. Similarly, sulfur get oxidized to sulfoxide and sulfone in our body. So by putting silicon, can we take care of those things? In addition to that one, can we push molecule more into brain? Why I am talking about that one? Because this, if you calculate C-log-P values, that increases C-log-P value, that increases lipophilicity, you can push molecule to the brain. That's what the advantage we wanted to do. In addition to all these things, maybe you are not able to see this one, there is a novel compound, you get patentable compound. That's the advantage with this approach. So again, this particular slide, what I want to show you is, so linezolid case, again, these brain infections are very serious problem, and if you, even for testing also, you need to go through this even for looking itself problem, there's lumbar puncture you need to do. It's a very serious issue. And some of the other ways also you can do, but this is the most ideal way to look at. And what we know at that time is linezolid already tested in patients with the BID, and they showed that they showed at least 6 out of 13 patients, 6 patients got cured. That information what we have proof of concept. So that's the advantage with this one. But most of the antibiotic don't cross blood-brain barrier. By putting silicon, can we think of doing that one? That's what we wanted to understand. What we did is we synthesized a compound with silicon. Not only just one compound, we made about 35 compounds with different groups changing here and there, other places. Then we collaborated with Dr. Rajesh Gokhale's team in IGIB, and what we found is actually 2 to 3, 4, like 4-fold less potent when compared to original compound. That's what we got. TB, not TB, yeah, TB also, and also other bacteria also. Less potent when compared to original compound. But our goal was to see whether molecule is going to brain or not. That's what our main goal. Although we can increase the dose and we can push it to the brain. That's what we wanted to do. So we collaborated with, not collaborated, basically we got grant along with Incozen. So just to tell you that I am the first grantee of CRS scheme. There is something called contract research scheme. Bayrak started. So this was one of the first project they did it. We got that one along with this Incozen. It is a Hyderabad-based company. So what I wanted to show you is what is the data I'll show you. This is the original molecule linezolid, and I want you to concentrate on this brain to plasma ratio. If you look at this Cmax or AUC, Cmax, let us concentrate. That's a 0.12. And by putting silicon, that Cmax became 1.2 brain to plasma ratio. That means like a 10-fold improvement. And similarly, other molecules also we have like a 25-fold and even 30-fold increase also there. That's really exciting result. Look at this one. This is a brain to plasma ratio. This is a original molecule, and this is silicon compounds. We have like a 10-fold, 20-fold, 30-fold improvement in blood. Like brain to plasma ratio. Although we compromised in some of the other parameters, but what we wanted to show is like whether by putting silicon, can we improve brain penetration, or can you push more molecule into the brain? That's what we wanted to show. We were successful in getting there. So that's what about silicon. We have many other projects under silicon. Right now we are doing in our research group. Okay, I also wanted to show you one slide. If somebody is interested, can take up this one. This is another company we collaborated when I was in NCL Pune. There's something called Shantani. So they have validated target, and we synthesized compound, and we patented this particular molecule. Again, I am showing this Marcus structure here, and we have one beautiful compound here, and we filed so many countries, and we could not basically out-license that company because they deprioritized this compound, and lot of data we have in this. Right now we have this anti-diabetic compound, some of the parameters also we have. If somebody is interested, I'll share the full data of this compound, and I'll connect with that company. So this is what we have, one compound, at least it is sitting with us right now. That company is no more interested, so we are thinking whether it's real to go forward or not, but maybe some of you can help us whether real interesting to move forward or not. So that's something we wanted to discuss. So I took little more time, and what we are trying to do is we work on application-oriented organic synthesis. We make molecules. Our job is only making molecules, but we connect with the appropriate biologists and other people who can create value for the molecules. More than that, students who work in my group, they get exposed to at least working towards the direction we're solving some problems. That's what we have this big team, like basically my team. I'm thankful to them because they made my career whatever I am. Whoever is there outside, they're all my first set of students, basically all who got PhD from me. Almost, I think about 15, 16 are already completed PhD. And right now present group is here, who are in IICT. And I'm sure one of you can recognize Remya. So she also worked with me when I was in NCL. So she joined here a couple of years ago. That is before I joined IICT. So again, we last few years we scattered different places, and basically Pune, Jammu, Lucknow, and Hyderabad. Now hopefully we'll continue to do some of the work what we have been doing, and hopefully we are okay with that one. So these are some of the funding agencies and collaborators. These are the people who made, and again my parent organizations, NCL, IICT, and Jammu Lab. These are the places we could create value for that one. Again, finally, I would like to thank you all. So I'm talking only as a scientist, no director on me now, right now. Okay? Yeah, if you have any questions or suggestion, I'll be happy to take. I know I took little more, actually more time, almost 20 minutes more. Yeah. Clearing. So see, silicon is only one atom out of whole molecule. So, but that our enzymes can recognize other parts of the molecule. If you go to other, I'll tell you one example. Yeah. See, if you take this molecule, the silicon is only one portion, one of the molecule, right? Our enzymes can oxidize any portion of the molecule. So the We don't know that one. At least I don't know that information we don't have. But what we do, how we get cleared is you get oxidase anywhere. With cytochrome 450 can oxidize any of these places, right? Once you make this hydrophilic, then you can push us out of the body. So that's advantage. We don't know even silicon metabolism also, at least I did not study, nobody studied, but that also possible. But based on other information, I can tell you that molecule get excreted, basically other places metabolizes and goes out. Yeah.
So for any of these silicon compounds has been done with PKPD?
Yeah, this is PKPD, now whatever I did.
So how much time does it take to be eliminated out?
Again, depends on compound to compound. It is also like any other compound. Some compounds may have better PK, some compound may not have. Again, depends on other part of the structure. I don't think anything really related to silicon PK. I don't think anybody studied.
It's also getting metabolized in the cell.
That we don't know. I mean, like we haven't studied that. We did not isolate that compound and studied that one. But I'm sure it can get metabolized also. If not silicon, at least attached compound like this methyl salts can get oxidized. That is also possible. We don't know that one.
Around the silicon, difficult to get metabolized because biological enzymes
May not recognize that one. So that is one of the basis also to put silicon there. Silicon will not recognize, whereas sulfur, our body recognizes sulfur, that gets sulfoxide and sulfur. By putting silicon, your enzyme may not come nearby that one. So that's one of the bases, but we don't know. That is only hypothesis. There is nothing proof right now. Yeah.
So based on your silicon, you are pushing your molecule to the brain, right? So should I understand that all the molecules were replaced with silica after carbon? Do they have tendency to go to the brain?
Yes, at least in this case we showed at least 4 or 5 molecules. In other scaffold also we showed. At least so far we have that last week we published, there is a tacrine. You all know tacrine? You can Google it and also see that. Tacrine.
It's the anticholinesterase.
Huh?
Tacrine. Yeah, it was the first anticholinesterase.
Yeah.
Cholinesterase inhibitor.
Yes.
That was being prescribed for, thought to be useful for Alzheimer's.
Yeah.
Discovered long time.
Yeah. The tacrine, what we did is we made silatecrine now. So that's just, it's out there. You can put it, Google, you'll get. Silatecrine, if you put, you'll get. So there also we showed that one. Because different places we showed. Again, there is no guarantee, but it's only.
See, some of the drugs which are there for Alzheimer's, like monoaminoxidase inhibitors and Parkinson's people who are using it. Do you have any idea whether they made any silicon approach for those things?
No, not much made, not many people made. Only few groups, whole world, only few groups are working. Only 1 or 2 companies are working and a few research groups are working. Because since it's a very narrow field, at least in India nobody is working, that much I can do. Right, Anthony? Anthony will know. He's a pseudo-chemist.
So in this particular approach, the silica approach, since as you said, it won't be degraded or recognized by the biological enzymes, doesn't that also act as a demerit when it comes to sustaining within the body in a longer period? So
No, why do you say test? No, it will not stay long period. In fact, actually this is a less stable when compared to other counterpart, sulfur counterpart. That means it is going out. It is not there in the body, right?
Okay, so it's basically getting degraded.
Yeah. Basically, that's what I was telling to madam also. See, need not get metabolized here. It can metabolize any place. See, what is a metabolism? We have to make molecule more polar and push out. Thus, our body mechanism is pushing out. How does it? How it push? Make polar compound, put hydroxy group, carboxylic group, whatever it is, using all kinds of enzymes. You put some polar groups there and push out, so that it will be more soluble. It goes out of our urine or species. That's the idea. So this can go same thing.
But then, as you said, this particular silica compounds are less druggable compared to its original ones, right? As you said, 70% it was less druggable, right?
Where did you say? Did I say something like that?
In one of this, it's not very potency compared to the original one.
No, potency is different.
Original one is less, but when you do replacing the silicon, it increases the potency. There was a graph. Yeah, that was the graph.
So, not this, previous slide. Previous, I think,
No, I don't have any other slide, silicon, only this. Maybe you are confusing with other one?
scale is what you showed, right?
Huh?
70%. Potency wise, you told one bit less.
Less, almost 3-4 fold less, less potent.
Okay.
So instead of, see, but where we have a compound is, like let us example. So one particular compound, See this compound I said, for example, this 9A compound you take, that is almost 30-fold more potent. So if you have less fold, 4-fold less potent also, we are fine, right? Then 30-fold you are pushing the molecule, right? Your more molecule, more compound is in your brain than plasma, 30-fold more. So that's our advantage. Yeah.
So similar to silicon, any other element you can?
There is something boron, already molecules are there, boron is there. Selenium also people use. Selenium, boron, what I know, few things. Again, deuterium is a recent drugs. And yeah, that's all we have.
And why only one silicon? Can we put more?
You can put then more silicon, means again you will tell me that there's so many silicon, what happens to body? No, I did not try, but in other project we tried. We can put silicon, more silicon. So initially we try with one silicon. If more lipo, you can increase. But again, how far you can go? Again, we need to balance it also. If you put more lipophilic, then the drug may not be absorbing also. You need to balance that a little bit.
Even sir, for silica-based different nanomaterials, there are reports that it is cleared through as by the formation of silicic acid, soluble silicic acid, and then it can interact with the different type of enzymes and it can make the metal enzyme soluble and it can clear. There are several reports are there for silica as nanomaterials used, and it is only compound. It will be definitely.
Okay. Yeah, I don't know much information in that.
Yeah, I have seen a lot of literature actually.
Okay.
Silica-based nanomaterial. That it will be actually converted into the soluble silicic acid. When it is soluble silicic acid, because in the acidic medium, they can make the silicic acid. So it is soluble.
Okay.
So once it is soluble, then it is easily clear. So that type of reports, many reports are there. For nanomaterials, silica nanomaterials, but it is not nano
Yeah.
material.
Yeah, it's not
So
nano.
It is anyway soluble in BMSO and any other solvent.
It is not nano, it is only macro.
What is the study about the shunting, what you mentioned about it is an anti-diabetic molecule.
Yeah, anti-diabetic molecule.
So why it did not see?
Huh?
Why it got stuck?
Why he got stuck means he spent a lot of money, that company spent a lot of money. So from NCL side we only contributed intellectually, but they put a lot of money, but they could not get the, I think that time only all these GLP compounds, other compounds came, gliflozins also came, that did not get into the, we could not license it, but he spent a lot of money. I feel bad for that person, but he spent a lot of money on that. Of course, he moved to different direction. Proteomics, Shantani Proteomics, that company, now they are getting into, initially they wanted to do drug discovery, they started and filed together a lot of patents. Of course, CSIR also spent a lot of money. We filed more than 50 countries, that interest he showed. So then he paid all the money.
So by any chance have you thought about using this structure for any drug repurposing? Any other efficacy?
Why repurpose? Purpose only you can use, no?
The purpose is a strain.
No.
It could not achieve, compete along with the
Other existing molecule.
So now,
Yeah, but
why can't you think about thinking something?
if you are interested, I'll give you that molecule.
You can look into if you can target or something.
Yeah, I'm sure. I can also tell, since it's a whole story, this
Diabetes is again a comorbidity along with the main disease. So one can see if that can be used as a for drug repurposing for any other target.
Yeah. No, I have more information, little target information also we have on that one. Actually, we wanted to write paper. He was excited to write to some nature chemical biology, and again he lost interest. So then he did not write, but I did not push him more because I was feeling bad that he lost a lot of money.
Now he loses the hope of chemical biology to be too bad.
Yeah. Okay.
Thank you very much, sir, for spending your time. Actually, by coming also, he's talking to the parliamentary committee. Okay,
Now I have to go and do that one. They are waiting there.
Students must have got interest and now you must have realized what is the strength of working here with molecules which are like
Good, use opportunity, interact. Student, not that faculty only talk to faculty. Students, you get interacted, and I think some like places there are use opportunities, not only just for only doing on the stage. You can also do labs. Also, you can interact. That's the advantage being in this particular place. Use that advantage We'll also connect with even CCMB also, we want to connect with them also. We're trying to do, in fact, one of the program we are doing, we didn't present, but there is also, for the first time, we filed a patent together. We're publishing one paper now, together with the CCMB and here. Antiviral.
Okay, I think we can all take a break, 5 minutes, and come back again for the next session.
Thank you all.